Good evening, everyone. I'm Prachi Pasalkar. On behalf of Pune Knowledge Cluster, I welcome everyone to this webinar by Professor Gagandeep Kang. We are very fortunate to have her here today for this webinar. And now I call upon Professor Ajit Kembhavi. He is the principal investigator of Pune Knowledge Cluster and a renowned astrophysicist. Over to you, sir.
Thank you very much, Prachi. So it's my privilege this afternoon to introduce Dr. Gagandeep Kang, who is one of the leading virologists, immunologists, and epidemiologists in India. Now, these words were all unfamiliar to many of us just a couple of years ago, but we keep hearing them all the time now. She's a professor of microbiology at the Christian Medical College in Vellore, where much of her research work was done. She was also the executive director of the Translational Health Science and Technology Institute in Faridabad during 2016 to 2020. The Professor Kang has been a researcher for a very long time, but her major research focus has been on viral infections in children and the testing of the rotavirus vaccine, which is used to protect against rotavirus infections. These infections are the leading cause of severe diarrhea among young children, and preventing that brings immense benefit, especially to the poorer countries. Dr. Kang has made immense contributions to our fight against the current pandemic through important committees of the Government of India and various state governments, and through the academia and popular media. Through her work, she has established strong training programs for students and young faculty in clinical translational medicine. Dr. Kang has received many awards and recognitions, including the Infosys Prize in Life Sciences. She is the first woman from India who was elected a Fellow of the Royal Society. And she continues to be the only woman in India to have the honor at the present time. She has served on many important national and international committees, which also constitutes a very important contribution on her part. Today, Dr. Kang will be speaking on prevention is health and wealth, which is a most topical talk we could hear in the current circumstances. She will tell us about the importance of vaccines and the great benefits that they bring, especially to the poorer countries. Dr. Kang.
Thank you very much for inviting me to join you here today. I'm going to be talking to you about vaccines. Obviously, they are something that has been front and center for everybody in the past few months. And the way I wanted to present this was from a lens of looking at prevention and the value of prevention. And this is critically important because the Pune Knowledge Cluster is a grouping of scientists, and I'm sure all of you are asked to communicate about vaccines, about COVID-19 in many, many ways to your community. But thinking about the benefit of prevention is something that the health community has focused on for the past several years. And this is what I hope to share with you today. So if we look at prevention, you can think about prevention as being upstream prevention, which is essentially the fact that where you're born, where you grow up, and how you live really determines what happens to your health status. You can also think about prevention in terms of primary, secondary, and tertiary prevention. And in primary prevention, we prevent disease and harm before they occur. And a key example of that is vaccination. Secondary prevention is when you know that a disease is likely to occur in a population. So you try to make sure that you find it as early as possible. So for example, doing screening for cervical cancer or breast cancer in women is a form of secondary prevention. You don't allow disease to become severe before you begin to manage it. And then tertiary prevention is really when you treat people appropriately, make sure that they can be rehabilitated, that the consequences of having had the disease are limited. So today we are really going to be talking about primary prevention, which is to prevent the disease before it occurs. And I've broken the talk into three parts. One is an introduction to vaccines. I recognize that there is a wide group of people here. So some of it will be very basic for many, but I hope useful nonetheless. I'll talk about the impact of vaccines on disease and death, and then about how we can place a value on vaccines going forward. So what really is a vaccine? It's a protein, a polysaccharide, a nucleic acid, a living vector, or an infectious agent, which either when injected or administered through the mucosa as an oral vaccine or a nasal vaccine or given as potentially a patch, can induce antibodies and cellular immune responses that are specific to the targeted pathogen. That response then results in protection against the pathogen, and that can be shutting down the pathogen completely or shutting it down sufficiently that it does not cause disease. Now, if we look at vaccine development, I think the newspapers wind up being pretty full of achievements that come from early stage studies where many things that happen in basic science labs are purported to be the next solution, either as a drug or a vaccine, because a molecule has been discovered that interrupts the pathogenesis of an organism or of a disease. But when we think about vaccine development in the academic space, usually the way it is approached is to identify the mechanism of natural protection, find the antigen or antigens that are responsible for protection, show in animals that the giving the antigen as a vaccine results in protection, find the best way to present that antigen. And also in some situations to learn from another pathogen to predict what might matter and design vaccines based on that. And an example of where this has been done was really in SARS-CoV-2, where we built on the work that had been done for SARS-CoV-1 and for MERS coronavirus to design the vaccines that we have today. Once you get through this academic phase, the vaccine candidate goes to industry, where industry figures out how to make it. And usually because they want to make lots and lots of the vaccine and show that it is pure and safe, assuring the quality of the product, they work out the manufacturing processes, they try to increase the yield and purity of the antigen, show that it has safety in animals, and then produce the first lot under good manufacturing practices. At this stage is when the vaccine comes under a regulatory ambit and then is taken through clinical trials. The clinical trials, the design of the clinical trials can be very similar, despite the fact that we might have many types of vaccines. So given in the next two slides are the various types of vaccines that we have today, with examples of licensed vaccines or approved vaccines that use those technologies and when we first got those vaccines. So an example of the smallpox vaccine is actually slightly different from a live attenuated vaccine, but it could be considered a weakened form or a different form of the smallpox virus that is given as cowpox to induce immunity against smallpox. So that was the first vaccine that we had. Subsequently, we have many vaccines that are based on either weakened or inactivated organisms, organisms that are not capable of replication, or if they are capable of replication, are not capable of causing disease. You can also have, in the case of many bacteria and viruses, a killed whole organism. And this is an example of this is the whole cell pertussis vaccine that is given in the DPT or pentavalent vaccine that's given to children. You can have toxoids where you take the toxin from bacteria such as the bacteria that cause diphtheria and tetanus, and then treat that toxin chemically so that it is no longer capable of causing disease, but induces a strong immune response. You can have subunit vaccines, which can consist of different kinds of purified proteins or peptides or polysaccharides. And these are components of the infectious agent that can be given to people in order to produce an immune response. You can have a range of other vaccines as well, virus-like particles, where you have a structure that resembles the virus, but is incapable of replication. You can have outer membrane vesicles, which are usually secreted by bacteria, but you can have strains that produce them in large volumes. And those can act as antigens. And this has been done for group B meningococcal vaccines. And then because generally polysaccharide vaccines do not induce a long-lived response, you can conjugate that polysaccharide to a protein, which results in your ability to make a vaccine that has a good immune response that persists for a long period of time, even against a polysaccharide antigen. We have viral vectored vaccines. Ebola was the first one that was made, and we have now got SARS-CoV-2 vaccines, which are viral vectored, Sputnik, AstraZeneca or Covishield, as well as the Johnson & Johnson vaccine, and many others that are in development. We have nucleic acid vaccines, both DNA vaccines from Zydus Cadila, the RNA vaccines from Moderna and Pfizer, and the vaccine that is under development at Genova in Pune. There are attempts now to make bacterial vectored vaccines, and there are experimental vaccines that use antigen-presenting cells. And there are 2 candidates against SARS-CoV-2 that are being developed by the Chinese army. Now, all of these vaccine candidates will undergo similar kinds of testing. Preclinical, there will be animal safety toxicity studies in India in 2 different animal species. If you have a good animal model, you can do immunogenicity, you can do challenge studies, all of which provide very useful information to vaccine developers. And then you take the vaccines through phase 1, phase 2, phase 3, and phase 4 studies. The numbers of participants increases at each stage, because the most important thing to remember is that when a vaccine is put into humans, the first thing that we want to look at really is this safe for us to be giving this to healthy people? In the preclinical development, a lot of characteristics get described. The manufacturing processes are set up, the quality control measures are defined. And as I mentioned, if there is an animal model, you can look at immunogenicity and efficacy studies. When you come to phase 1, this is done in healthy adult volunteers, and you're looking to see that there are no serious or unexpected effects of this vaccine. And this includes the dissemination of either if you have a live vaccine or you have a nucleic acid vaccine, you want to make sure that it's not going anywhere else except where you gave the vaccine. This is usually done in a small number of subjects, and these studies usually don't require a control. In phase 2, you go into target or high-risk populations. Here you begin to use controls. And usually in phase 2, we do lots and lots of different kinds of studies to determine the dose, how it should be given, whether it can be given with other vaccines, and how it response is in the presence of pre-existing antibodies. There is a whole lot of evaluation criteria that you have to go through. And some of the discussions that people are having today about the gaps between doses is because we didn't do the usual number of phase 2 studies that are done to determine dosing schedules in most of the vaccines that we have today. In phase 3, you go into target populations, and here you start to test the vaccines in thousands. In the case of the SARS-CoV-2 vaccines, we tested in tens of thousands, and this is usually a controlled trial, and you can use either a placebo or an active comparator. Here, the goal is to try and figure out, does the vaccine protect against the disease that we want it to protect against, and how well? So making sure that you have your case definitions is important. You can also do additional studies that look at consistency across different batches. You can do bridging studies, and in India for vaccine licensure, these are done a lot, where you say that essentially if you make an antibody response that is similar to an antibody response that is made to another vaccine, then if that vaccine has efficacy data, then we accept that this vaccine will too. Phase 4 studies are done post-licensure, and these can look at duration of protection, look at very rare safety effects, as well as how the vaccine performs in real life or in subpopulations that have been given the vaccine. Now, when we look at how vaccines are performing, mostly we look at vaccines in what are informal phase 4 studies. And the impact of vaccines can be phenomenal. I'm showing you here the example of 2 vaccines. The first is a Hemophilus influenza type B vaccine given in the US, where you started out with a polysaccharide vaccine. It reduced disease a little bit. But as soon as the conjugate vaccine started to be used in older children and then in younger children, you could see that the rates of Hib meningitis just crashed. This is what vaccines can do directly to the disease that they are targeting. If you look at the next set of figures, this in the red is the use of a vaccine that is directed against Neisseria meningitidis A. You can see here that the non-A Neisseria are not affected at all. But as soon as the vaccine is introduced into a vaccination program, and this is in sub-Saharan Africa, the meningitis A just disappears again. And this was a vaccine that was made in Pune. This is Menafrivac, which was made by the Serum Institute of India. And within a couple of years of the vaccine introduction, essentially the epidemics of meningitis A that happened in sub-Saharan Africa just stopped. So that is, to some extent, the effect of vaccines on diseases that the vaccine targets. What do routine immunization programs do? And there has been a great interest in this because when we first started out, vaccines were used as a tool to prevent deaths, and specifically to prevent deaths in children. As the number of children dying has gone down, policymakers have been more and more reluctant to introduce new vaccines because the number of deaths that are prevented are relatively low. So recognizing this, Gavi, the Gavi Alliance, which is responsible for introducing vaccines in low and middle income countries, established the Vaccine Impact Modeling Consortium in 2016 and asked them to bring together the world's best modelers to look at 10 diseases that are vaccine preventable, to look at it from diverse points of view, to build models to predict what was happening in terms of deaths in places where vaccines were or were not introduced. What they did differently from what had been done before is that all of the modelers were asked to use the same demographic data for 98 low and middle income countries and use the same vaccination coverage data. And the period that they were asked to model was from 2000 to 2030. This consortium was established in 2016, and the idea was that there was real data for 15 years. And could the modelers use this real data for 15 years to predict what the next 15 years would bring? And these data show, these figures show the vaccine coverage in 2000 in figure A, in 2010 in figure B, and in 2000 and, sorry. And then in 2019 for figure C, and in figure D is shown all the vaccines that were introduced into low and middle income countries at various points and what the vaccine coverage was during that time. So as you can see here, some of the vaccines started to be introduced very, very late. So the modelers had to work with data. For example, for some of the vaccines that were introduced after 2010 and 2015, like rotavirus and human papillomavirus, they had to use figures that were just being generated at the time or came from high-income countries. And when they did these studies, they found that for 8 out of the 10 vaccines in the birth cohorts that they were studying, children born during those years, they could see that vaccines continued to prevent death in those birth cohorts, except for hepatitis B and for human papillomavirus. And this makes sense because the HPV and HPV deaths that you are preventing will actually be deaths that are prevented for birth cohorts that would have been immunized much earlier, or you had to predict for a time point that went out beyond 2030. If we look at deaths averted by disease and by country, the key findings are that individuals who are born in 2019 will experience 72% lower mortality due to the 10 vaccine preventable pathogens over their lifetime than they would with the counterfactual of no immunization. And without vaccination, all-cause mortality among children younger than 5 years would be 45% higher than currently observed. So even though deaths have declined a lot, as shown by the global burden of disease estimates, vaccines are still continuing to prevent mortality and will continue to do so until 2030 and beyond. So what is it that we expect from vaccines? Obviously, we expect from vaccines that they prevent death and they prevent clinical manifestations of disease. But if we look still at the effects of vaccines, what we would also like vaccines to do, and some vaccines to do this, is to get protection against asymptomatic infection or colonization. And when this happens, that means that you have a reduction in transmission and possibly herd immunity, which means that you're protecting more individuals than just the vaccine recipients themselves. Some vaccines may also drive changes in responsiveness to infections with different pathogens, and these are called non-specific effects. And this may be happening because you're stimulating prolonged changes that occur in the innate immune system. And this is something that is under study for people who have been vaccinated with BCG vaccines in early life. Now, in herd immunity, I thought I would put this slide in because herd immunity and its absence or presence for SARS-CoV-2 is a question that is frequently discussed. And this is a nice figure that shows what happens. So here, if you have a person who is sick and a population that is entirely susceptible, if you look at the panel on no vaccination, that infected person can then spread the disease to all the susceptible people in the population, so that pretty much everybody in the population gets infected. Now, if your population is made up of people, some of whom are susceptible and others who are protected through vaccination, then an infected person coming into that population will be able to infect the people who are susceptible, but will not be able to infect the people who are vaccinated. There will still be some people who do not come in contact with an infected individual and therefore do not acquire disease. This is what happens when the vaccine coverage is below the threshold for herd protection. When the vaccine coverage becomes very, very high, then you've reduced the number of susceptible people in the population, and therefore the chances that an infected person will come in contact with those susceptible individuals becomes greatly reduced, and infection cannot easily spread. So susceptible individuals are indirectly protected by being in a population of highly vaccinated individuals. So I'd like to come now to some of the work that we have done on rotavirus, and I want to bring this as an example of what we will discuss next, which is what is the full value of vaccination. Now, when we started out working with rotavirus in India, there was a huge resistance to introducing the vaccine. And the reason was that people said that rotavirus is a disease that can be very easily handled by the healthcare system. All you need to do is make sure that a dehydrated child reaches a hospital and they can be rehydrated at the hospital if home-based rehydration did not work. There is no specific treatment for rotavirus, and we are not sure that there are sufficient numbers of deaths in this country that require for children to be vaccinated with rotavirus. We think that it would be more important to vaccinate against something else, like possibly the pneumococcus. So we did a whole lot of work trying to establish the burden of disease for rotavirus, and we were able to build the disease pyramid that you see at the bottom of the slide, essentially showing that at the time that we did the study, which was in 2011, 1 in about 350 children born in India died of a rotavirus infection. And this alone wasn't considered sufficient for introduction. So we went ahead and also tried to look at what were the other consequences of rotavirus diarrhea on children, trying to identify whether it resulted in children becoming malnourished, We also looked to see what rotavirus infection meant for households. And quite frankly, it came as a shock to me even though I'd been working in the community for a really, really long time, to understand that a rotavirus hospitalization of a child for 3 days resulted in 5% of the median household annual income. And this was a median expenditure. So the number of people that can be driven into poverty because of what seems like a relatively minor illness in children was just incredible to me. And in studies that have been done around the world, if you look at this bubble chart that is shown here that shows the deaths averted. Deaths averted for many diseases like measles are very high numbers, but for other diseases are much lower numbers. And yet, if you look at the costs that are incurred by families on treating children that have these diseases, you understand that prevention by vaccination can result in families not being impoverished. If you look at India, the World Bank figures for India tell us that 63% of all expenditure on healthcare in India in 2018 was out-of-pocket expenditure. A lot of this goes on hospitalizations, about 1/3. A fair bit goes on pharmacies where money is spent before people wind up in hospitals. And it's estimated that about 60 million people get pushed into poverty because of these catastrophic health expenditures. Catastrophic health expenditure being defined as more than 10% of household income being spent on health. So I started then to look at vaccines and what vaccines could do for society and not just for the prevention of disease. And if we look at vaccines and the benefits that we get from vaccines, we can have the narrow perspective, which includes the healthcare cost savings that come to the healthcare system as well as to households. We can look at care-related productivity gains, or we can have a broader perspective where we look at a lot more areas where prevention of disease and prevention of death by vaccines can result in an impact on individuals, on families, and on societies. So to fully capture the value of vaccines, there are a lot of things we need to look at. What can immunization do? Immunization promotes equity. It results in reduced morbidity and mortality. It can result in herd immunity. It results in better child survival and therefore lower fertility. Parents don't have to stay home to look after their children. Children who are not sick can go to school. This results in improved cognition and education. There is lower healthcare expenditure if you prevent disease, and that results in more money being available for increased consumption. All of these ultimately result in a situation that really changes what happens in society. You have a demographic dividend, you have more money available in the system. That results in an increased tax revenue and potentially increased GDP. All of this sounds a little bit incredible when you start from a health perspective, but if you actually begin to look at data, there is data to back this up. Vaccines, in fact, can impact other diseases. It's been shown that in patients with diabetes, if they had influenza vaccination, that resulted in a reduced risk of all-cause death, as well as cardiovascular disease. There are also examples of where you can change what happens in terms of finances. We know that diseases drive people into poverty, and vaccines represent something that promotes equity, but also protects poor households from catastrophic health expenditures. There's been a modeling study that looked at what the impact of scaling up measles, rotavirus, and pneumococcal vaccines would be in low and middle income countries, that showed that unequivocally being able to vaccinate children meant that there would be less catastrophic health expenditure. In addition to that, many of the vaccine preventable diseases that we work with are infections that have serious sequelae and can result in lifelong damage. Hib and pneumococcal infections are examples of that, where it has been shown that survivors of Hib and pneumococcal meningitis have very severe cognitive impairment, in addition to other neurological sequelae. It's also been shown, not just for Hib and for strep pneumoniae, where you can prevent the meningitis, it's been shown for measles that measles vaccination can have an impact on educational attainment of children, and that has economic consequences that extend beyond early childhood. There are also synergies that can happen between health programs. Families who access the health system for non-vaccine healthcare use are more likely to have fully immunized children. So non-vaccine healthcare encounters can serve as opportunities for vaccination and vice versa. And this is something that most health systems are looking at. Vaccines can also reduce antibiotic use, and their potential impact on antimicrobial resistance is something that is being examined now. This paper on the effects of vaccination on invasive pneumococcal disease in Africa showed that not only did you reduce disease, having a pneumococcal vaccination program resulted in a 16% reduction in antibiotic usage in those communities. And in places where pneumococcal vaccines had resulted in herd immunity because of sufficiently high coverage, that amplified reductions in disease and also reductions in the use of antibiotics. Vaccination also reduces pressure on the healthcare system. Here is data from a paper that showed what happened when pneumococcal conjugate vaccine was introduced into the program. There was a reduction in the need for antimicrobials. There was also a reduction in acute otitis media, and therefore the need for the healthcare system to do tympanostomies. So to end, vaccines have clearly transformed public health. If we start from smallpox, the first and so far only human disease to be eradicated, or look at any of the vaccines that have been introduced into vaccination programs, as shown in the graphs on the right for data from the UK, you can see that vaccines have really made a difference to infectious diseases around the world. There is a need to invest in vaccines, not just for the impact that they have on disease, but for the impact that goes well beyond that. So one way of looking at it, and circling back to the title of my talk, if you have vaccines contribute directly to Sustainable Development Goal 3, which is good health and well-being. But if you look at it, if you had immunized and healthy children, that would result in a productive future workforce, that would result in stronger economies, and that contributes to goal 1, which is no poverty. So prevention is important for health, but prevention is also important for wealth. Thank you very much, and I'd be happy to take questions.
Thank you so much, Dr. Kang. It was a very inspirational and a lot of information was shared. Thank you so much. So it's open for questions now. Participants, you can raise your hands or can put your questions in the chat box.
I have a question, please.
Yes,
Hello?
yes sir, you're audible.
Yes. So how much would it cost to get the whole world vaccinated for the kind of diseases that you mentioned? Because after our population is only 7 billion. And what would be the cost? And would it be possible to meet this cost in the present circumstances without affecting the economy?
So it really depends on how many diseases you're vaccinating against and where. So in high-income countries, those countries already have the resources to be able to vaccinate their populations. And now the problem is really in middle-income countries where vaccination rates are sometimes lower than in low and middle-income countries, because in low and middle-income countries, there is Gavi or donor support for vaccination. So if we look at all of the costs of routine immunization, currently, leaving out SARS-CoV-2, we give about 4 billion doses of vaccine around the world. And it's been estimated that even though those are not the prices that are charged, almost every vaccine that we have can be made for less than $1 a dose. Now, if you think of each vaccine requiring an average of 2 doses, you're talking about potentially 8 billion being the cost of vaccination around the world for non-SARS-CoV-2. For SARS-CoV-2, it's been estimated that what we need, if we don't go into children, is about 11 billion doses of vaccine. And the average cost of a SARS-CoV-2 vaccine is $6. And that needs to be given as 2 doses, so $12 to fully immunize a person.
Thank you.
Thank you so much, ma'am. So we have a question by Ravindranath ji, and he's asking that there are quite a many videos by serious immunologists suggesting that vaccines against COVID do not prevent transmissionability, and that the public health policy agencies such as CDC do not bring out this feature. So they are saying that.
Okay, so I get the question.
Yeah. Okay. Yeah.
So one, of course, nobody should be forced to be vaccinated. But if you are working in a setting where you want to prevent infection, then your employer can ask for you to be vaccinated. So for example, I have no problem with a healthcare facility saying that if you want to work in healthcare, you must be vaccinated, because you cannot be a risk to your patients who are in any case vulnerable. Now, the question of transmissibility, actually, for the first 3 months after vaccination, the data very clearly show that the mRNA vaccines reduce transmission. It's less with the Delta variant, but protection against transmission for the first 3 months is high. After that, it drops. With the AstraZeneca vaccine as well, there are studies that show that there is a reduction in transmission. The most recent study that was just published in the Lancet, which shows that people who are infected with the Delta variant, whether they are vaccinated or unvaccinated, carry the same levels of virus, still show that the period of shedding is shorter in vaccinated individuals, which indirectly translates to some reduction in transmission. So I don't think that you can say that vaccines abolish transmission. They do not, but they do result in a reduction in transmission, either directly, which is what happens in the first phase, or secondarily, which is what happens later when the people are infectious for a shorter period of time.
So we have a few more questions, but sir, would you like to go first?
Yeah, may I ask a question? Dr. Kang, my question is about the recent discussion on mixing of various kinds of vaccines. Without going into the politics or the judgment about it, are there any data about it? Is there any other disease in which vaccines of different origins have been mixed effectively?
So for Ebola, the Janssen & Janssen vaccine uses 2 different platforms. One vaccine is a vectored vaccine and the other uses modified Vaccinia Ankara, an MVA platform. So the antigen is delivered in 2 different ways. One is through sequence and one, yeah.
So, because
it has been,
it's an advantage, is it?
Yes.
Okay. So it is possible that if sufficient data become available, it could be used in other diseases also, prevention of other diseases.
That's correct. I mean, so I think one thing to understand is that no vaccine that we have today is perfect. Right. And no vaccine is completely safe. So why would you want to mix? You would want to mix to improve the performance of the vaccine or to make it safer if you give a combination. Particularly if you have a side effect with the second dose, then don't give the second dose, give another vaccine. Those are the kinds of approaches that people are thinking about now.
Okay, thank you very much.
One more question we have from Priyanki, and she's asking that how do you think the development of the COVID vaccine will impact the overall time from the lab to vaccination of individuals going forward the vaccines?
So I think COVID was an unusual situation where you had no limitations of money and you had a regulatory system that was very facilitatory that resulted in vaccines progressing very fast. What we are now finding out is that even though the COVID vaccines went through phase 1, phase 2, phase 3 studies, all of them, actually we kind of abbreviated the phase 2 phase, which is where you would test the dosing, where you would test the intervals, where you would test combinations. We didn't do all of that because we were in a hurry to get to phase 3. So now we are coming back and we are doing all the other studies that will allow us to define how we can use vaccines most appropriately. Certainly, we've learned lessons from this, that it is possible to move faster. If we absolutely have to, we can. Will this result in a change for every vaccine in the future? That is a little difficult to say. It would depend on the extent of, how important is the disease in which part of the world and how much resources available to develop that.
So we have next question by Priya Nagraj. Do you want to unmute yourself and ask Priya? Okay, so maybe, yeah.
Thanks, Prachi. Yeah, can you hear me?
Yes, we can hear you.
All right. Good evening, Dr. Kang. My question was that so there have been some reports about an ideal rate of vaccination, and if not done at that rate, we would have a smaller group of vaccinated people amongst a larger group of unvaccinated people. And my question is, does this affect how the vaccine, how effective the vaccine is at the population level?
So if you look at, so let's look at individual protection, right? First, individual protection depends on when you got vaccinated and mounted an immune response. There's a small amount of variability in that, but once you've received your first dose for some vaccines and both doses for other vaccines, you will be well protected sometime out after you have received the vaccine.
Right.
Now, the rate of vaccination in a population matters if you are in a closed community.
Okay.
Right? And in that closed community, if you want to get to herd protection, you would like to cover as many people as, particularly when the force of infection is high, you want to be able to protect as many people as fast as possible.
Okay.
Right? Otherwise, it doesn't really matter.
Yeah.
You do it as quickly as you can.
Okay.
In some situations, you may want to go. So in routine immunization, what we do is we cover one birth cohort at a time. When a child becomes age eligible, we vaccinate the child. We have a schedule for that.
Right.
There are other situations where there is an outbreak, for example, where you want to go for campaign mode immunization. And there the goal is to try and protect people as quickly as possible and, if possible, shut down transmission.
Okay. Okay, thank you.
Thank you so much. We have one more question, if we can take that. Can we take it, Dr. Kang?
Sure.
Yeah. So we have a question by Sanjay Kanvinde. So he also has a comment and saying that PKC is bringing different domains and institutes in Pune ecosystem to address the challenges in health sector. Thank you so much, Mr. Sanjay. And his question is, Dr. Kang, if you look 10 years out, what new opportunities do you see for acceleration of innovation in primary prevention domain?
Well, ideally, I'd like to see social determinants of health addressed, but that is significantly more challenging than developing vaccines. And I think, you know, why I wanted to do this talk from this point of view and not focus on a single topic was really to bring home how important primary prevention is, because really prevention can change the face of society. So if we think about what the Pune Knowledge Cluster can do, thinking about vaccines and prevention through vaccines, what is the maximum benefit we can achieve? How many diseases can we target? We know that our immune systems are very powerful and can mount protective immune responses against many, many more antigens than we give today. So how many more could we protect against? And what would the bang for the buck be for each additional disease that we targeted?
Thank you so much. It was a very informative talk, and especially the question answers and the discussions after that was very interesting. Thank you so much, Dr. Kang, for accepting our invitation and joining here today for the webinar. We were really fortunate to, get to hear you today. Over to you, sir. Kimberly, sir, would you like to say something?
I just want to give a big thank you. And this has been, I've never had the opportunity to hear Professor Kang speak before this. So this was a great experience for me. I'm sure it'll also be a great experience for other people. And I only request that we can interact more with her to learn a little bit from about what we could do through the PKC for epidemiology. Thank you very much, Dr. Kang.
Thank you. Bye.
Bye.
I'll end the webinar.